This presentation will be given at the Brazilian Medicinal Chemistry Conference on November the 11th, and it's regarding how we are serving the medicinal chemistry community with Cheminformatics platforms that we're developing at the Royal Society of Chemistry. All the slides, as usual, will be available on my SlideShare account with the URL above. The RSC has been a provider for chemistry for many years as a charity, as a publisher, as a host of commercial databases, and I will touch on a couple of those, as a partner in grant-based projects in recent years. I will certainly mention some of the work that we're doing there. We are the host of ChemSpider. Many of you will have heard of ChemSpider. It is a very large database of chemistry data that is freely accessible to the community. And we're also now developing something called the RSC Data Repository for chemistry. I will cover all of these today. So the situation is that our scientists are getting increasingly overwhelmed with data. There's no issue really anymore with generating the data that we might need to examine for our own chemicals. Certainly if we're searching for information regarding other chemicals that are online, there's so much online data available today. If you compare what's available now with what was available 10 years ago, it is incredible how much of a change we've seen in terms of online data. Organizations are releasing data, pharmaceutical companies, National Institutes of Health, so much data coming online, and much of this is available to us to use as we see fit, and certainly to support efforts around medicinal chemistry. Of course, funders are now encouraging openness to the point where they expect people to have data management plans and be able to share their data once the research is complete and put it out for the community to use. This is only going to increase, it's only going to go alongside the shift towards open access publishing. One of the things that we do is lots of data is used, downloaded, it's modeled for specific purposes, and then commonly those models are lost. Even in publications when models are reported, there's no real easy way to access the training set, the validation set, the model that was produced, so that it can be used ourselves. So what would be great is that there was a platform where models could be put for the community to use. And actually, this is mostly not a technology issue because these types of platforms already do exist. Adoption of the platforms, utilization of the platforms, getting people to share their models there, that is a bigger challenge. Pharma companies repeat a lot of work, certainly in the domain of using online data. So much of this data is downloaded by pharma companies, biological data, screening data, chemistry data, protein data, enzymes. There's so much out there, and most of the pharma companies download the data, they put it into their own databases, they integrate the data, they analyze it, and they create their own solutions inside an organization. And this just goes on and on and on and on in pharma companies, and it's just a lot of repeat work. And there have been changes in this in recent years, and I'll mention one of the projects that is challenging that and going after a better solution. Publications, of course, lock up data while they release it in consumable formats such as PDF files, and for those still printing journals, that in that form also. The reality of it is that when I read an article like this, there is no easy way to download the data from these curves for me to save, reanalyze, repurpose. The molecules are all there as sketches, and while some of the publishers are making it available so you can download it as mol files, etc., in general, publications are locking up all of the data, and it would be much more powerful for us if we can access it all. So I'm writing this article right now regarding proton N15 long-range shift correlation with my friend Gary Martin, and there's some beautiful images going in here. These are actually structure files with signed chemical shifts, and if we could pull these data out and put them into databases for free release and make sure that the article itself was indexed and structure searchable, and the correlation information could be captured and shared, all of this would be very powerful. But of course, most of that is going to be lost too. What if we could actually navigate? So what if a medicinal chemist was reading an article like this and wanted to ask some specific questions? Or if they wanted to download the compound so they could perform searches against their own databases? What if they wanted to look at pharmacology data that may be sitting somewhere else in some other database and they wanted to access it directly from an article? There are a whole set of questions, some of them shown here on the right-hand side, that a medicinal chemist could ask, and that would be more of an ideal path forward. However, let's not lose hope, because there are efforts underway. There are certainly platforms being built that should help in this domain, and I want to cover some of those right now. So ChemSpider is something that should be well known to the majority of chemists at this point. It's got over 30 million chemicals. It's been collated from various data sources, over a few hundred data sources at this point, and people supplying data directly to us, data that's available online. We have allowed crowdsourced curation and annotation. We get data from our own journals, and what we tried to build was a structure-centric hub for searching the web. We've achieved that to a very large extent. I will review some of that. And I also want to point out that it's become a really, really big dictionary, and that is important for some of the things I'm going to discuss. So let's take a look at ChemSpider. We do a search on levothyroxine. It's the biggest selling drug in the United States. This is the chemical compound. We see here masses and systematic name, etc. We have data here that can be used for searching. So if you have monoisotopic mass, of course, for a mass spectrometrist, that would be of value. If we go further down the record, we find a set of names and identifiers. These are all actually bolded. That indicates that they have been validated by experts or by appropriate robot engines that we have developed around certain rules. And there are a lot of other names that have not been validated, and if people want to check out that record, go ahead and just do the search. We have predicted data online. These are the data from ACD Labs Perceptor platform. We also have experimental data here, and you can link out to predict properties on the ChemAxon site. We have literature references, so we do searches of the chemical names that we captured, those validated names, and we hit the appropriate APIs to look up the data on RSC journals, RSC books. In this case, I'm showing us a hit list on PubMed, but if you want to see the full list of articles, you can click out. And here we are searching Google Books. So what we've done is just pass that list of names over. We've also got patents here that are linked out, and this is onto the Google Patents site. And RSC databases, so if you want to know about mass spec analysis of levothyroxine, or analytical abstracts, where there's chromatography, etc., then these are just click-throughs to those articles, to those abstracts. And we also have the places where you can source the chemicals, and if you look under the vendor and data sources, you will find a list where they're hyperlinked. They will be highlighted in blue, and they go off directly to the vendor's website, and there you can source it, figure out from them cost, availability, etc. And with structures and names against these records, so levothyroxine has a whole set of chemical names, and we have the chemical structure. Of course, we can do things with it, such as searching further across the ChemSpider database by substructure. We can also go off to external sites, and we use a lot of the Google services for that. So here's an example of name searching. I've searched Google Scholar. I want to look only at articles since 2014, and you can see that here that we've passed out the chemical name list, and it's brought back a set of articles. And because every chemical we have on ChemSpider has an associated INCHI, and the INCHI is this International Chemical Identifier, most of us are using it on our databases nowadays. We're increasingly using it on publications. Then it is possible to search just directly onto Google, and here we see links onto various databases, ChemSpider, ChemID+, Brenda, Enzyme Database, etc., etc. So what we've got now is we've got a database of chemicals with powerful searching integrated to APIs across the web for names and structures. We've literally made the web structure searchable. What did we learn as we built out this database of tens of millions of compounds from various sources? We certainly learned that data quality is an enormous challenge. And we've done as much as we can to clean up the data. We still continue to to develop new ways to analyze and improve the data. And we also determined that getting the crowd, scientists who use the site, to annotate and curate the data has helped a lot. And this is just one example of an article that we've written. So it is possible for anybody who used the site to leave feedback, top right-hand side, on every entry. And we've had many tens of thousands of edits made to the site. Software can actually help, so it is possible to look at the massive data sets and push them through platforms to validate the quality of the data. We've done some of this already. We continue to tweak the rules as we move forward. And we can also standardize the format of the chemical structure diagrams according to rules. And we're following the standardization rules issued by the Food and Drug Administration in this particular document. The platform we've built is available for anybody to use. It's called the Chemical Validation and Standardization Platform, CVSP. It is available at this hyperlink, and you can upload one or many chemical compounds in an SD file, for example, and it will issue warnings and tell you where there are stereochemistry missing, charge balance issues, hypervalency, etc., etc. So feel free. Anybody can use the site, and it is good for validating your data sets. Now, ChemSpider is actually a building block, and when you layer API calls on this, provide web services, then we can build other platforms around it, and the community can integrate to it. And we have many sites integrating to ChemSpider already, even some of the analytical instrumentation vendors such as Agilent, Waters, Bruker, Thermo, etc. So with these web services, API calls available, and some componentized widgets that we built, we can use the ChemSpider data and some of the searches to support grant-based services. So we have done that, and we're involved in a couple of significant grants, as well as a couple of open drug discovery projects. And I just want to mention a couple of them. We all know the warning about the fact that the antibiotics are going to be a problem in the future as we become antibiotic resistant. We hear this increasingly now. And this is just one post that came from the BBC about the antibiotic apocalypse. We also know that over half the drugs introduced between the 40s and early 2000s were either directly natural products or were inspired by natural compounds, so the key scaffold for those pharmaceuticals with natural products. So with this in mind, there is a project called PharmaSea, S-E-A, as the spelling, so that we're trying to find new antibiotics deep in the ocean. And this project involves trying to harvest new materials extracted from the ocean and trying to identify what these compounds are, and looking specifically for novel chemistry. This is the actual website if you're interested more in the project. It's the PharmaSea. It's funded as an FP7 project out of Europe, headed out of the Aberdeen University, and involves a consortium of European-based scientists. So what we've done is we've taken a number of components that we've been building. I will mention them again later, but they're the chemical registry service specifically, and it utilizes the chemical validation standardization platform during the registration process. We've also built some widgets to design websites quite quickly so that we can set up a number of these with their fairly short time scale. And what we have here is the PharmaSea website. We have the simple search, so text-based searching, we have structure searching, and we have some advanced search capabilities I will show you shortly. What we are loading onto here is a marine natural product data set, because PharmaSea is focused on marine natural products. They don't need the entire 31 million chemicals from ChemSpider that comes from all over the place. They want to focus their searches in on marine natural products. So it's a much smaller data set at present, but just over 25,000 compounds. What we're ultimately trying to do here is develop this as a dereplication platform so that when new compounds are isolated, when compounds are isolated and they want to figure out whether they're new or not, they can use analytical data to search the database that exists of marine natural products. They can search on the mass, they can search on spectral features from the NMR spectra, so they will have number of methyls, the number of acetyl groups, features that they can easily identify within the data that they've generated. Then they can search these data against the overall data set and figure out whether any of these compounds have been seen before. It's called the replication process. And this is we've just released this for testing to the consortium. This site is not public at present, but ultimately some of the capabilities will certainly be made available. We've also worked on a platform to serve up the chemistry aspects of the project on something called Open PHACTS. Many of you will have heard of Open PHACTS. We do know how to spell Open PHACTS. It's not F-A-C-T-S. It's PHACTS, P-H-A-C-T-S, which stands for Pharmacological Concept Triple Store. It's a semantic web project. It works with a very large consortium of people. It's a 3-year Innovative Medicines Initiative project. That project has completed, and we have an extension to the funding to continue work. It's been very successful. We're integrating chemistry and biology data, specifically open data in general, and using semantic web technologies to build out the platform. And there are academics working on this, multiple pharma companies engaged, and publishers, not just ourselves. And the overall premise is to make sure that we're using open standards, open data, and producing open code that can be reused during the lifetime of the project and later, so that ultimately the data can be downloaded and brought in-house at the pharmaceutical company. It's not just the pharma companies, anybody can do this, and we'll be able to provide an instance of the Open PHACTS platform that can run on internal servers. So when I was talking before about pharma companies doing repeat work, this is an example where the pharmaceutical companies realized that they were indeed ingesting a lot of the public domain data, bringing it together, and just doing so much repeat work that they got together, worked with the Innovative Medicines Initiative to support this particular project and get the funding, and I would say it's been very successful. This slide is already well out of date. There are so many more companies involved in the community at this point. I just don't have all of the latest logos on it, but it shows you indicatively, I hope, of how many groups are involved. So again, we've been working on the chemistry aspects of this. So this is the Open PHACTS website, ops.rsc.org, is available for anybody to look at. Primarily, we've served this up just to look at the chemistry for our own testing of what we've been producing for the members of the Open PHACTS consortium to look at. But it's very, very limited to the chemistry. But it does have advantage. So the entire Open PHACTS data set is available as an open data set from the Open PHACTS website. It can be downloaded. Here you can do searching. You can do structure-based searching, similarity searching, substructure searching, text-based searching. And because we have INSHIs generated for every member of the database, each of the compounds, you can take advantage of things again like the INSHI searching. So in this case, where you come to the INSHI key, you click on the on here, and you can search this exact structure. So for this compound, we find 5 hits on Google, and then you can navigate out and read about it, and you can see that we have a source of how to buy that compound. We have it on ChemSpider a couple of times. We have it also sitting over on an NCBI database that we'll go straight through. So what about the biology? Well, this is where the actual work done regarding meshing together the biology data and the chemistry data, which is a very complex piece of work. This is the actual architecture that we're adopting. There's biological expertise within the consortium that is working on bringing the data together. Our own chemistry expertise. We have people who have high expertise in linked data, the semantic web. We work with the Virtuoso Triple Store, and a lot of this requires that we work with RDF from each of the data sources. We're also looking at things like nano publications, which you may have heard of, that we can actually host, and you can see it over here sitting in this particular part of the database. It's a lot of work. I won't go into all the details on the technical side of this. It's its own presentation, but our aspect has been dealing with the chemistry normalization, quality control, and generating the RDF from the data sets to mesh into the semantic web databases. So here we see the Open PHACTS Explorer, which is one of the paths into looking at the particular data sets that we already host. We've got about 2 million chemicals sitting inside Open PHACTS and all the associated proteins and enzymes, etc., etc. I'll show you some more examples of this shortly. But you can use this. It's open for everybody to use, so it is an open project. Everybody can go and play with it. Some examples. In this case, we're doing a particular search on a compound. We've linked into this from an article. I will show you how that works later. But we've pulled up the chemical, and we've got some very basic information about it. We've got pharmacology associated with this particular compound, and there are 2 particular hits here, and you can look at the assays that have been measured. It is possible to do searches based on compounds active against a particular enzyme family. So there are 6 enzyme families that are listed here, and you can drill down into this and look at the particular enzyme family and each of the particular aspects of how that is segregated up by the enzyme classification. And here we show 2 of the hits. There are a total of about 200 hits in here, and you can see the data. And it can all, of course, be downloaded as a file, viewed in Excel, etc. Here's an example of doing pharmacology by target, so that you can select a particular target that you're interested in, and it will retrieve the chemistry. So this type of meshing together of data, of course, is dependent on access to a lot of open data, and as you may know, the linked data sources are growing enormously at this point. This is an old slide with old numbers, generated in 2011, so things are moving very quickly, and there are billions of triples are out there at this point. What we've had to do on ChemSpider is ensure that we can make the data available in RDF format. That is certainly what we've done. So ChemSpider itself is fully available in RDF, but the Open PHACTS data set is about 2 million chemicals, and we've made that available, fully open data in RDF form. And we've got the pieces that we've been building for this, the chemical registry service and the chemical validation and standardization platform, are available as open source code, released to the community, and will be they're available from GitHub. Right now, the chemical registry service is available from GitHub. It will be updated shortly, and CVSP will be another component. Going back to the fact that ChemSpider is a dictionary, the fact that we've got all of these systematic names, trivial names, the fact that we've generated INCHI strings, INCHI keys, and have various registry numbers and database IDs, means that we have this enormous dictionary that we can utilize. And as an example, if we look at Valium or diazepam as a synonym, we see many chemical names on here, some systematic, we see some of the trivial names, and when you have this type of dictionary available to you, you can make some very, very interesting connections. So as an example, the RSC, we have many articles, and what we've been working on is semantic markup of the articles, originally called Project Prospect. We now call this prospecting articles or semantic enhancement. The intention is when you're reading a journal article, a chemical name tells you very little about the actual compound. So in this case, we're building interfaces where you hover over the name and it shows you the chemical structure. But in reality, what we want to do is go even further. And once you've got the chemical structure, we take you out to the ChemSpider platform, where we can store a lot more information about that compound, of course. And as I showed you, from here we would have links to patents, links to other publications, links to other databases, and you've got all the structure searching and text-based searching that I showed you via the Google calls. So that's been a start point, but we're providing even more links in recent work that we're doing specifically for the medicinal chemistry community via our MedChem.com journal articles. And we have been working on this for a while now, and this is very much early version, but you can see the ability to display compounds, chemical terms, biomedical terms. You switch these on or off in the interface. This is on an HTML page, and you can see it highlighted. Here's an example of scrolling further down the page. We're reading about all of these chemical compounds, and we're interested specifically in one of these chemicals, and here it is. Istradefylline. Hover over the name, and now you're allowed to do more with it. And what comes up is the ability to migrate out to ChemSpider as before, and view an image of the compound so that you can insert it and utilize it in publications, presentations, etc. Because we've been working on all the Open PHACTS platform, then we have the chemistry queries linked up for Open PHACTS, and you can even push it out and predict toxicity on the open platform, ToxPredict. Here's an example of just coming out from MedChem.com directly into ChemSpider for that particular compound, and directly out to the Open PHACTS Explorer for that particular compound. So this is with new articles, and MedChem.com is one of our most recent journals, but we have a lot of data, of course, sitting in old publications, and we would love to bring out as much data as we can from there and use it for various purposes. So we can certainly build it into databases. If we have a lot of data, then we can model that data and make it available, make the models available, make the data available for other people to build models. We are thinking of ways to extract the figures from the articles and database them and link them back, as well as finding analytical data. And I'll show you some of the work we've been doing of late in this particular area. So our archive goes back to 1841, and we have a lot of articles, over 300,000 articles sitting in the archive. Of course, in the supplementary info, we have so much information. We have, in this case, a synthesis. We have the synthesis diagram. We have the synthesis text. We even have the analytical data written in text forms, and there are ways to do things with that. So any chemist will be able to look at this particular couple of paragraphs and be able to see the chemical names. So of course from chemical names can be used to generate chemical structures, and this can be done algorithmically. So if we can extract chemical names, then we can certainly convert these into chemical compounds, assuming the names are correct, assuming that the people used correct systematic naming approaches. We can do that. And don't forget, we have a very, very large dictionary in ChemSpider, so we can actually pull out trivial names too and start to link them back to the articles. So we can, in theory, make our entire archive structure and substructure searchable and linked up to the database. There will be a need for curation, there will be a need for further validation, but we have a chemical validation standardization platform to deal with that issue. So all these components are starting to assemble together to form a very, very nice story for what we can do with chemistry data. Of course, the actual way this is written up, the context tells us about the reaction, and it is possible to actually extract a chemical reaction, not just the chemical compounds, but an actual reaction from this. And we've been doing work with Daniel Lowe at NextMove, who has been doing mining of Google patents to extract chemical reactions. And this is an example of a reaction that has been extracted, and it shows below the actual part of the text from Google patents. If we can take the same approach on our archive, then we will be able to pull out all of the reactions and make them available in a reactions database for searching. Of course, with every chemical linked, and as much as possible, we would like to extract analytical data too. So I showed you the text form of the spectra, and this is a very common representation. This is a carbon NMR spectrum. We can all read this and understand it. It means that it was running in deuterated chloroform, 100 megahertz resonance frequency, 400 megahertz system. We've got a list of shifts here with multiplicities, and essentially we've got a good description of the spectrum. This is also true for proton NMR, of course, and we've been working with Mestre Labs, a Spanish company, to take their tool that takes textual spectra and converts them into visual depiction formats that scientists would probably be more willing to compare with an experimental spectrum than a list of shifts. So this is generated directly from the text spectrum. It's a virtual spectrum. It will not be identical to what was generated on the spectrometer, but certainly very useful in terms of whether you want to do data validation and comparison. We're also going through and taking figures out of our journal articles and our supplementary info. There's an NMR spectrum. The figure at the top is the real spectrum. The one at the bottom is the extracted spectrum, and I think you would agree that there is very little difference between them. There are, of course, differences, noise levels, and some of the satellites are missing. You lose some of the smaller couplings, etc. But at least we can make a lot of progress here. In reality, the raw data made available and posited with us at submission time so that we can hold it in a content holder like ChemSpider and link back as supplementary info is a much better way to go. But that depends on the scientists providing us access to the data and allowing us to capture it and share it via our databases. Of course, all data can be embargoed until the publication is issued. We can also host data for many other publishers. There's no reason that you can't put analytical data, spectral data, NMR, infrared into our database and link to it from other publications. That's absolutely acceptable because data cannot be copyrighted, and having live data links is a much superior way to go relative to locking it up in PDF files. Because we're extracting data from using the text mining approaches, we're also going after things like melting points, boiling points, log p-values, aqueous solubility. This is an example of a model built on a very carefully curated data set of melting points that was done with work with a gentleman called Jean-Claude Bradley, who worked on open drug discovery. So this model is an excellent model. It's probably the best model that's ever been generated on a melting point data set, according to some of the comments we've received back from QSAR modeling people. This is the model that was built, and we started a search against Google Patents, and this is extracting melting points directly from Google Patents. It's only 170 records, but you can see very, very good consistency between the model and the extracted values. The data extraction has continued. We have close to 100,000 melting points at this point extracted, and we're presently going through the data doing some filtering. There are some obvious errors that are not in the extraction process, but actually obvious errors when the melting points were written up into the Google Patents. So we're doing some filtering, and we'll be reporting on this and publishing in the very near future. So 100,000 melting points, close to. Again, flash points are coming out, boiling points. With boiling points, we have to take into account, you know, at what pressure were they measured? Did things could decompose, etc., etc. So working on text mining, what we'd love to do is also to carry text mining directly over to theses, because there's so much data sitting in theses that never gets published. In fact, I recommend, my comment is that for all of the data that I've generated, maybe 5% of it was ever published, maybe 15% of it was ever in my thesis. So I've lost a lot of data. And there are things we're thinking of doing about that. I'll mention this a little bit later. But right now we're trying to get the chemistry data out of theses. In order to build a national compound collection, as we call it in the UK, we worked with some students, funded some students to extract data from theses. So the intention was pull the data out of theses, build a library of compounds, approximately 50,000 compounds extracted, do some virtual screening, identify whether there were any compounds of interest, and then since they were all extracted from theses, in theory, the physical samples may still be available in the universities in question, or because they're reported in theses, then the actual synthesis is there, and we could actually synthesize some of the compounds and do actual screening to see whether we have any interesting responses. We built this also on ChemSpider, and we put the data in the supervisor and the university code, and we built a template for the data entry, and many tens of thousands of compounds are in there, and the work is progressing. So that's the National Compound Collection. However, as I said, there's so much data that's actually not published, doesn't even end up in theses. What we would love to do is build a place for chemistry research data management. So we're building out an RSC data repository. It's much more all-encompassing than ChemSpider. It'll have containers for compounds, reactions, analytical data, and we will have algorithms in place to validate the data on the way into the databases. We will index and search as appropriate, because each compounds versus reactions versus analytical data require different search technologies. And of course, we will do our best to model the data that we get available and build predictive algorithms. So this is chemistry research data management, not just abstracted data. So this is just some early screenshots of what the data repository looks like for compounds, for reactions, for analytical data. These are all live interactive data. When available as open data, they can be downloaded and reused, etc. And crystallography data at a very basic level, just holding CIF files and making them searchable. So with all of this data in hand, sitting in the data repository, with the data that we presently have available through the Open PHACTS platform, and of course the data sources continue to expand, with what we're doing with making the articles more enhanced for searching and prediction directly off of the article, as I showed in MedChem.com, maybe it's time to envisage that these articles will be much richer. You will be able to do much more with them. Sure, the PDF file is still a great way to distribute the data. However, we believe that there will be a lot more linkages coming off of the articles, a lot more data directly available from the articles, and you will be able to navigate and perform research in a wholly different way. So conclusions from what we've been doing and what I've presented, we have certainly been building platforms that we believe can support the MedChem community, but not only. We've been working on supporting a lot of other communities, and we will continue to expand our reach into things like the materials science community, etc., the analytical sciences communities. We are certainly working hard to extend our data, but we're very selective at this point. We want to make sure that we try and keep the quality of the data up to spec, because the general quality of online data has been suspect, and a lot of our investigations have urged caution. We're of course opening APIs to the platforms, and when we've done this, it provides a really powerful building block to build around these the data and the searches and services. You've seen some examples of that. We have many others. And we're also open sourcing components of our platforms to the community, so that people can work with us and build their own systems, and you'll see that increase over the next couple of years, we hope. Hopefully this was of interest, and here are my contact details if anyone would like to get in touch. Thank you.